In this video, we'll spend a few minutes to look at the cellular extensions. Now, when we're talking about cellular extensions, these are formed by the inner framework of the cytoskeleton. And the three examples are the microvilli, the cilia, and the flagella. So when we're looking at the microvilli, what we're looking at are these finger-like extensions. We can see them illustrated here on the artist diagram. They're these finger-like extensions, and they also have a core of actin filaments that help to basically give them structure and help to maintain their shape. Now, of course, one thing you'd notice quite quickly is with these microvilli, you can see how much the surface area of the cell is expanded. And not surprisingly, where do we find microvilli? We find them in areas where we really need a lot of absorption. So, they show up in places such as the GI tract, other parts of the body that we'll see as we go through the course. So just remind ourselves, so an extension of the plasma membrane, there's an actin filament core, and of course that brings us back to that core principle of structure and function go hand to hand. Whenever you see microvilli, you can almost pretty well bet that we're looking at an attempt to increase surface area because I need to absorb materials at that place. And as I said, as we zoom in, you can see, look how much surface area there is. That really gives a lot more surface area. And we do have those actin filaments that run up through here. Now, let's move on to the next one, which are the cilia. And the cilia are these hair-like projections. And what are they made of? They're microtubules, plus they have motor proteins. Now that last bit, motor proteins, gives us an idea that there's movement involved, and there is movement. Now don't think of cilia as being like windshield wipers on a car that just go back and forth. It's a very coordinated movement, and it's about moving material in one direction. So in order to do that, they have to work in unison to move substances. So they're going to move substances along, up and out. And one great example is many of the cells in the respiratory tract are ciliated to move mucus up and out and secretions up and out from the lungs. And you can see what happens is, if you start here, let's say we had, let's make it green, some green mucus here. So if I had some green mucus to move up and out from the respiratory tract, you can see what happens is the cilia at one point are standing fairly straight up. And then what they do is they bend over, and as they bend over, they're going to move the material along. Okay, and then any given cilia, once it's sort of gone through this process of bending over, so this is trying to show us what's happening with one cilia. And so as the cilia bends over, it's going to move the material along. Okay, as the material gets moved along, then what happens is the cilia has to get back to its original position. So this is what it looked like when it starts the cycle. This is where we want to get back to. So how does it do it? It uses these motor proteins in the internal structure to bend over. And as it flops over, then what it does, it has to do, so we often call this the power stroke. Then it has to do the recovery component. And then what it does, instead of just being straight upright and then going this way, almost like windshield wipers going back and forth, that would defeat the purpose. If this was straight up here and move back this way, it would likely move the mucus back to where it was, and we're back where we started. So what it does is it flops over using motor proteins. That's the power stroke. And then the recovery stroke, you notice that it doesn't stand straight up. It stays sort of flopped over. And in this position, it returns. You can see that it returns to its shape. So there's the cilia. And as I said, it's really about moving things over the surface of cells, a group of cells, or in certain tissues. The next one are the flagella, and flagella we don't see that often because they're really only found in sperm cells. They're solitary, they're longer than cilia, and they move in a different type of fashion. They sort of a whip-like fashion that propels the entire cell. And now the one thing is flagella and cilia are structurally similar to centrioles, except they have these two central microtubules that you do not find in centrioles. And we can just have a quick look at that. You can see this looks very similar. We have these groupings of microtubules out here, but in addition what we have are these two ones in the middle here. And as we sort of finish, as says, you finish off every little section of the book, it usually comes back with a summary, and we can see some of that summary right here. Talking about cilia, flagella, and then it gets into a number of sort of disorders, which you should have a look at in your book and look through in your notes. Okay, I'm just going to leave it there, and that was just a quick introduction to things like the microtubules, the cilia, and the flagella.